Hello, everyone. I welcome you at our GeneSprout webinar series titled Plants as Grandparents, Doctors and Chefs. First, I would like to thank the American Society for Plant Biologists, ASPB, for hosting our webinar series. We are very grateful for this opportunity. This webinar series, Plants as Grandparents, Doctors and Chefs, is recorded and will be available on the YouTube channel of the GeneSprout Initiative and of ASPB. Today's three talks will focus on plant health and their medicines. Our three speakers are young scientists from the GeneSprout Initiative. Our first speaker is Prisca Meyer, who will be talking about the fight against plant diseases. Second, Gwen Swinnen will tell us all about how plants defend themselves against these diseases. And third, we have Barbara Berckmans, who will go deeper into molecular biology of these plants' own pesticides. I am Vince Verbeke, and I will be your moderator for today. I am a PhD student in plant biotechnology at Ghent University, and I am a proud member of the GeneSprout Initiative. I will guide you through today's three very interesting talks. So I would say, sit back, relax, and enjoy the science. After and during each of the different talks, there is an opportunity to ask questions. You can post your question using the Q&A button on your Zoom window. We will have a general Q&A session after the three talks. First, before we start, I will give a very brief introduction to the GeneSprout Initiative itself. The initiative is a collective of young plant researchers based in the Netherlands and in Belgium. And with GeneSprout, we try to promote an open dialogue about new plant breeding technologies and their policies. As an organization, we try to organize discussions, workshops, and debates on this topic. As for instance, we had a campaign before the start of the global pandemic called Give CRISPR a Chance, in which we try to inform the broader public about the CRISPR-Cas technology and its potential applications in agriculture. We are, of course, on social media. If you type GeneSprout on Google, you will find us on several platforms. I will also place some links in the text box to these different platforms. If you ever want to contact us, please do not hesitate and send us a message to one of said platforms. Now I will give the floor to our first speaker, Prisca Meyer. Prisca is a promising last year PhD student in bioscience engineering at the KU Leuven. She was born in Germany and obtained her joint master degree at the Technical University of Munich and the Humboldt University of Berlin. Her current PhD research in Belgium focuses on vertical farming of strawberry. She uses LED lights in different conditions to control plant development. Today, she will be talking about strawberries and gray mold rot. Welcome, Prisca. Please start sharing your slides.
Yes, thank you, Vincent, for the nice introduction. I think you can all see my screen. I hope so, at least. Okay, so welcome also from my side. And now welcome also to the first talk of today's webinar series. And we talk about fighting plant diseases on the example of strawberry and gray mold rot. But before we start, I would invite you to imagine some very delicious strawberries for a second. Probably, or hopefully, you imagined something like this. So very nice red strawberries that you have at home or that you just bought at the supermarket, or even some strawberry desserts like the cake you can see here at the bottom. But this is not the reality, but this is. So we can see diseased strawberries in the field, but also a package that we just bought in the supermarket. We brought at home, we wanted to open it to eat them, and then we found there's already some disease going on. So while we grow strawberries for a long time already the Greeks and the Romans cultivated strawberries, we have yield losses up to 25% every year due to different diseases. And one of the disease is gray mold rot. This is the disease that you can also see in the images. And this is caused by a fungus that is called Botrytis cinerea. If I would translate that literally, it would mean grapes like ashes. And if you have a look at the images, you can imagine why it's called like ashes, because it has this really gray, weird texture on the fruits. And it's called grapes because it was first found or described in grapes. So this fungus causes the most destructive strawberry disease worldwide, the gray mold rot, but it can also infect more than 200 other crops that we are growing. But the most interesting thing about this fungus is, at least from a scientific point of view, is that it's quiescent. So what does that mean? Normally, you can see in the image, the spores of the fungus, they infect ripe fruits like the strawberries, and then the disease breaks out. But the spores, they can also infect flowers and leaves. And actually, the natural way of infecting, or what we can see in nature the most, is that the spores infect the flowers and then they stay quiet or quiescent. That means only when the fruits are developing, or only when we harvest the fruits, the disease breaks out. And now you can imagine that a disease that you can't see is very, very difficult to control. So how do we control it at the moment? We, of course, or what we mostly do is we can spray fungicides, so chemical control, but this has already led to some resistant strains, and it's always in the talk to impact the human health and, of course, the environment. We can also do post-harvest control, so we can store the strawberries in low temperature conditions or in a controlled atmosphere, but this has, well, the controlled atmosphere causes off flavor, and the effect of the low temperature is very limited. So we desperately need some alternative methods. And one of this methods could be UV light. A short excursion, what again is UV light? So UV light is the part in the sun that causes the sunburn, or if we get too much of it, can even cause the skin cancer if we don't protect ourselves against it, as the woman is doing in the video at the moment. UV light is actually divided in 3 parts. So we have the UVA, the UVB, or the UVC part, which are the wavelengths that are shorter than the visible light that we can see. Actually, the shorter the wavelength, the more harmful and destructive the part of the UV light is. So the UVC light, for example, can directly damage our DNA. But luckily, UVC light is not reaching our Earth due to the ozone layer. However, UVA light 100% comes through, and around 5 to 10%, depending where you are in the world, of UVB light reaches our Earth. And it affects us humans, as we know and as we have seen, but interestingly, it can also affect the plants. So here you can see a leaf of a plant that just grew under light, under normal light without UV spectrum. And here a plant that grew under high UVB radiation, and you can visibly see that there's a difference in the leaves. We are now working not with the whole UV spectrum, but with only UVB light. And probably you're wondering, why did we pick out the UVB radiation, the one in the middle, and not UVA or UVC or the whole spectrum? And this is because I think most of you probably know is that the plant has different light photoreceptors, which enable the plant to see different light colors, or they can perceive the light quality. So here are some examples. We have the cryptochromes and the phototropines that enable the plant to see in the blue light region. We have the phytochromes that enable the plant to basically see in the red and far-red region. But interestingly, we also have a UVB photoreceptor, which is called UVR8. And this is very interesting because once these photoreceptors are triggered, they trigger further downstream pathways and genes. So that means if we give different light to a plant, like extra UVB light, we can trigger everything that's connected to this photoreceptor. And the effect that UVB light has on the plant is actually very, very immense. So I have some examples here, but they are just examples. There are thousands of effects on the plants. So UVB light influences seed development. It influences how the leaves are growing. So the leaves can get smaller, they can curl up and get wrinkled. It influences the height of the plant, but also the branching. So you can see that this plant under UVB light produced more side branches. It influences the growth of the plant, but also the stomata closure, meaning when the stomata are closed, of course, the plant can do less photosynthesis, so it has less energy to use. But it can also influence the lateral root development, even if the UVB light does not directly reach the roots. However, we have to take into account that it always depends on how much. So it's as with us humans, if we go out in the sun for 5 minutes, this doesn't harm us. If we outside at the sun, in the sun for 5 hours, our skin gets red, and the same is for plants. So a little bit of UVB light doesn't harm the plant, but a lot of UVB light can stress the plant immensely. But next to this broad effect that UV light has on the plants, there's one very interesting effect, and this is it influences the immune system of the plant. So it can make the plant stronger and more resistant to pathogens and pests. It also influences the amount or the composition of polyphenols, which make up actually a great part of the immune system of the plant. Short excursion to what are polyphenols again. Polyphenols are responsible for the taste and the smell of many plants that we eat. So I'm very sure all of us or all of you have already consumed some polyphenols today. We can, for example, find hydroxycinnamic acids in coffee and salad. We can find tannins in wine and fruits, but also in tea. Very typical, the proanthocyanidins and the anthocyanins in fruits, but we can also find some catechins in chocolate, for example, or we all know the typical smell of the onions. And with that, we come to the research question that I'm actually working on partially in my PhD. And this is now, can we use this UVB light? So can we exploit this UVB light to protect our strawberries against gray mold rot? Because we have learned that UVB light can trigger the plant immune system. So can we use it as an alternative method to fight this very destructive strawberry disease? And for that, I'm going to show you what has been researched before. So here you can see 3 different types of strawberries, Japanese varieties, that grew under normal light. And here we don't talk about the gray mold rot, but we talk about another fungus or fungal disease that strawberries get. And this is called powdery mildew. You can see some pictures here. And if plants grow under normal light, you can see that between 20 to 50% of the fruits can get infected by powdery mildew. However, if the plants grow under additional UVB light, this reduces to 0 to 7–8%. So that looks very promising. So is there some research that has been done with botrytis? And there is, but it's not on strawberry, but on the model plant that's used in plant science, the Arabidopsis. You can see some pictures here. You can also see that these plants that grew under control, so normal light, and these plants grew under additional UVB radiation. And there is a small difference in size, but it's not really striking actually. But very interesting is the result here. So in this experiment, the lesion diameter that the fungus causes in leaves was measured. Lesion diameter or the lesion area is what you can see here. So the brown part is basically what the fungus infected in the leaf, and this is the lesion. And if the plant just grows under control light, we have between or around 28 square millimeters of infected plant material. But if there's additional UV light, this reduces to half of it. So that also looks very promising. But it's of course never that easy, and it's not that straightforward. And this is what this research shows. So here you can see an experiment that was done on UVB light on strawberries on a biocontrol agent of plant pathogens. So basically, we again have the botrytis, the fungus that we don't have and that's want to have, and that's causing the destructive strawberry disease. But they also had a look at Clonostachys rosea. That's a fungus that you can also find in strawberry. And this is a good fungus, a positive fungus, because it competes with botrytis for nutrients. That means, and it's not harming the strawberries. That means if we have Clonostachys, we have less of the botrytis infection or less chance of getting the gray mold rot. So we want to keep the clonostasis on our strawberries. And here in the results, you can see with increasing UVB light, so here we see 3 different doses, we can see that the incidence of the fungus in leaf discs, so just cutouts of the leaves, increases for botrytis. This is not what we want to see. And at the same time, the fungus that we want to see is reduced. And on the contrary, we can also see the area with conidiophorus. So what was conidiophorus? If you remember the picture from the beginning, here we can see botrytis, and basically the stems where the spores are growing on, these are conidiophorus. And they are, with increasing UVB light, are increasing as well. This is not what we want to see. And at the same time, the positive fungus that we want to keep, this is reduced. So this result shows us that it's not that easy and not that straightforward. So what is the challenge that we are working on at the moment? This is that we want to give UVB light to the plant because we have seen that it can trigger the immune system of the plant and reduce disease incidence. But we can also accidentally give too much UVB light and then the plant is stressed. And we don't want that because a stressed plant also produces, for example, less fruits. And this is not what we want. So we don't want to harm the plant. So we only want to give a little bit of UVB light called supplemental UVB light. And here we try to find then the balance between damage and protection for the plant. And this is what I am in my PhD partially working on, or in general, what the status of the research on UVB light is at the moment. And well, thank you for attention. I hope you enjoyed the first presentation, and I will give the word back to Vincent.
Thank you, Prisca, for this presentation. It was actually very nice to learn about these things. I was quite shocked when you blew away my thoughts of these delicious strawberries with gray molds. It wasn't that nice, but the talk was very interesting. So I'm rather happy to hear that UV light could actually be used to combat these pests to protect our strawberries. So thank you.
Thank you.
Now, before I go to our next speaker, just a small reminder, whenever you have a question, you can pose it using the Q&A button on the bottom of the Zoom screen, and we will have a Q&A session in the end. Now, our second speaker is Gwen Swinnen. Gwen is a postdoctoral researcher in plant developmental biology at the University of Lausanne, Switzerland. She was born and raised in Belgium, where she obtained her PhD degree at Ghent University. During this PhD, she shed light on how tomatoes produce their own plant-made pesticides to defend themselves against pests. Now, since Gwen likes chocolate very much, her move from Belgium to Switzerland, where they also have amazing chocolates and beautiful Alps, was a very logical one, of course. So welcome, Gwen. Please start sharing your screen and share your story with us.
Thank you, Vincent. Let me see. Okay. I don't think this is working. Let me try again. Can you see my screen?
Yes, but maybe best if you can go now into this swap
presenter
screen,
mode.
So we see your presenter mode.
Yeah, the weird thing is that I don't really see anything. So I'm not sure. I only have like the little...
On the bottom of your screen, there was this this button in which you could click on this presentation. Maybe go for it. And otherwise, it's okay if you do it as such, I think.
Yeah, I think I know what the problem is. Sorry.
No problem.
Let's try again.
This is the disadvantage, of course, of these online meetings. It's always a bit difficult.
Yes, indeed. Okay, I think we are there. So what are you seeing now, the presenter mode or?
Now you have to indeed switch. Now we see
Okay.
presenter mode.
Now we're there. Good.
Okay, thanks, Gwen.
Nice. Good. All right. So again, thank you and hi everyone. My name is indeed Gwen and I'm a Belgian plant researcher currently working at the University of Lausanne in Switzerland, and I'm really happy to be here with all of you. And today I would like to tell you about some of my favorite pesticides. Now, first things first, what defines a pesticide? Like, what is it? Well, it's actually any chemical that independently of its origin, it can be natural or synthetic, so any chemical that deters or kills an organism that acts as a pest. So that can be snails, bacteria, viruses, caterpillars, and insects. Now, you might be surprised to hear that we consume about 1.5 grams of those pesticides on a daily basis. That corresponds to the mass of 137 sugar cubes per year. Now, I don't know about you, but I think that's quite something. Now, you might also be surprised to hear that only a mere 0.01% of that intake comes from pesticides found in crops, originating from treatments performed by farmers. Now, this is actually an estimation based on data from the Food and Drug Administration, so the FDA in the US, but data on pesticide residues gathered by the European Food Safety Authority, or EFSA, each year suggests similar estimates could be made for EU consumers. Here you see which level of pesticide residues were found in about 90,000 samples of EU food gathered in the year of 2018. So as you can see, in a little more than half of the samples, no residues from pesticides used by farmers were detected, meaning that either they just weren't present or they were, but at a level below what can be detected. In about 43% of the samples on the right side, residues were detected, but below the legal limits. And those legal limits, they are based on the toxicological reference values for each pesticide used by farmers, and also on the expected amount to be found if the treatment was performed correctly. That means that less than 5% of those 90,000 samples contained residues above the legal limits. So you might be wondering, where does the other 99.9% of the pesticides we consume come from? Well, they are pesticides that are made by the plants themselves, the ones that we eat. So in fact, in your lifetime, you are estimated to ingest about 5,000 to 10,000 different plant-made pesticides and their breakdown products. But why do plants produce these? And how come there are so many of them? Well, because let's be honest, our diet isn't that varied. But it's because plants are pretty amazing. Every plant species produces its own set of pesticides to fend off their natural enemies. And today, I will show you some examples of these, of what we call specialized metabolites, because every plant species has evolved, produce specialized chemicals that help them combat the specific pests that they are faced with. But before I go on to show you these examples of plant-made pesticides, let's have a look at what defines their specificity for and toxicity to certain pests. The first thing is their chemical structure. While one compound might affect a biological process in one organism, say an insect, it might not in, say, bacteria. And while one chemical might affect a biological process in 2 different animal species, this might be toxic to one of them, but not to the other. So you see all of these different chemical structures here. This is what defines that. Now, the second important determinant is the concentration of a chemical, which influences the dose that an organism will be exposed to and whether or not it will lead to a toxic response. This is nicely illustrated in this typical dose-response curve. And each chemical has its own specific dose-response curve, which shows that the dose of any chemical makes the poison. And this is no different for pesticides. And neither for chemicals we don't typically consider as toxic ones, like plain sodium, which can be toxic at a high dosage, and so is vitamin D, for instance. So these things explain why many of our beloved plant-made pesticides aren't toxic to us, but they are to certain other organisms, like caffeine, which is found in coffee beans and tea leaves, keeping us focused and awake, but paralyzes and kills many of the insects that have a nibble. Another plant-made pesticide we've come to know and love is vanillin. Even though we use it to add flavor to some of our favorite foods, vanilla plants produce it because of its antifungal and antibacterial properties. And next to knowing this as a natural chemical, you might also know it as a synthetic one, which is used much more often than natural vanillin, but it's exactly the same because the chemical structure is the same, as you can see here. So the plant-made pesticides that we consume on a daily basis do not seem to be harmful to us. Moreover, and counterintuitively, many of them are even beneficial to humans at the concentrations they occur at in plants. In fact, they can act as phytonutrients, which are plant-made chemicals that help prevent chronic diseases such as cancer, cardiovascular disease, and chronic lung disease. And one of them is chlorogenic acid, which belongs to the polyphenols that can be found in strawberry, which Prisca already mentioned in her presentation. But it is even more abundant in solanaceous species, such as tomato and potato, both very popular all over the world. And because of that, it's one of the most consumed polyphenols and also one of the most consumed pesticides, which is a good thing, since it's a very potent antioxidant that suppresses oxidative stress, and thereby it helps prevent cardiovascular disease. Now, in plants, it also works as an antioxidant, but in addition, it helps them defend themselves against chewing insects. Now, quite some plant-made pesticides can even be medicinal when given to patients at concentrations higher than they naturally occur at. So next to providing their own treatment when faced with pests, they can also provide treatment for us humans. So I'll show you two examples here. The first one being artemisinin, found only in Artemisia annua, which is specifically grown to extract artemisinin from, because artemisinin is the most effective anti-malarial drug. It kills the parasite that causes malaria and that is spread by mosquitoes. Now, this is, of course, not why the plant produces it. It does so in order to fend off insects. And as a last example, well, I'll actually show you two, which are two very closely related chemicals called vinblastin and vinclustrin, which are specifically produced by Catharanthus roseus, or you might also know it as periwinkle, to get rid of chewing insects. That's why the plant produces it. But we extract these compounds because they are also very potent anti-cancer drugs. They are used to combat different types of cancer, such as breast and thyroid cancer. And that's actually everything I wanted to tell you about today, which has hopefully shown you how truly amazing plants really are. And I would like to leave you with the following request. With your next meal, please take a moment to appreciate all those wonderful plant-made pesticides on your plate. Thanks so much for your attention, and I'll see you later when we answer some of your questions.
Thank you, Gwen. I'm very happy to have heard you speak. Sometimes you would forget that plants, of course, also make pesticides. I think it brings nuance to the debate of pesticides in agriculture, actually, in general. I also saw some interesting questions popping up, so we can answer those later. Now, again, if you have questions, please keep posing them in the Q&A box, and then we will answer them later after the next talk. So finally, Our last speaker is Barbara Berckmans. Barbara is a very talented postdoctoral researcher at the KU Leuven. Her current research focuses on the function of plant defenses during plant defense responses and root development. So as Gwen, Barbara was born and raised in Belgium, where she obtained her PhD in plant biotechnology at Ghent University. After this, she moved to Düsseldorf, Germany, where she was a postdoctoral researcher at the Heinrich Heine Institute, a university. 2 years ago, she returned to Belgium to join the plant-fungi interaction lab at the KU Leuven. So welcome, Barbara, and please start sharing your screen.
So I think it's fine like this. Yeah. Okay. So hello, everybody. Yeah, I will tell you today on how we could use plant compounds, not only for agriculture application, but also for medical applications. And for this, I will use plant defensins as an example. So the webinar of next week will be talking about the nutritional value of plants to humans. But it's not only we that see plants as a rich source of nutrients. Many other organisms do, like bacteria, viruses, fungi, or insects. And here I'm showing you only a small representation of the devastating effects that they come with by causing infections. And for us, it can have large implications as they go hand in hand with a large loss in crop yield every year. And some have even large impacts in history, like for example, the potato late blight causing the Irish famine and the massive migration towards America. Now plants, they do not have an immune system comparable to animals. Nevertheless, they have developed mechanisms which allow them to detect invading organisms and also to stop them before they're causing too much damage. And very broadly, they can be divided into physical barriers and chemical compounds. Physical barriers, you can see them as a first line of defense, such as, for example, the specialized structures like trichomes, which you can see here, and you can compare them with little hairs on your skin. Otherwise, plants have on their tissue above also some kind of waxy layer, which you can see here, the little purple threads, which has a waxy composition, and it makes it quite difficult for pathogens to invade. Other possibilities that plants have, for example, upon detection of pathogens, they can close down their stomata, which limits the entry points that pathogens do have. Now, next to this, plants also have a wide range of chemical defense mechanisms. For example, plants can deplete nutrient availability for the plant pathogen. Additionally, they can produce these specialized metabolites, of which Gwen was already telling you about, like for example, caffeine or citronella, which we all use also in summer to repel mosquitoes. Besides that, plants, they also have, or they produce a large variety of antimicrobial proteins. And there are really a lot of them, and they have a huge variety in functions, and it would take us too far to describe all of them. But today, I will be talking about the plant defensins, and they are actually one of the largest family of small antimicrobial proteins that the plant have. Now let's first do some small history lesson. So the first defensins were isolated from wheat and barley. And this was done in the beginning in 1990s. And 5 years later, they also discovered some similar proteins in radish seeds, and it was from them that they were named plant defensins. And from then, plant defensins were found in all plant species. And plant species can have a huge variety of them. So for example, wine, as you can see here, they have like more than 70, but you have also plants that have more than 300 or even more than 700 different plant defensins. So there is one thing that characterizes these defensins, and that is their protein structure. So as a reminder, so proteins, they are composed of amino acids, which are linked to each other like a chain. And depending on the nature of these amino acids, this chain will fold together into a three-dimensional structure. And this creates the protein structure. And for plant defensins, it looks like this. And we will not go or discuss this very much in detail, but what you should know that it's a very stable, rigid structure, and it's held together by very strong connections between the different amino acids. Now, this specific structure, together with the biological activity of plant defensins, gives them some unique advantages. So as first, among all the different plant defensins that were already studied, we can see that they have a very broad antifungal and antimicrobial activity. So when you apply some plant defensins towards a culture of fungi, you will see that you inhibit and you will almost kill all fungi cells within it. Another interesting aspect of them is that they're non-toxic to mammalian and plant cells. And why is this? It is because plant defensins, they bind to specific elements within the fungal cell membrane. And these elements are not present in mammalian or plant cells. Moreover, the elements that these plant defensins are binding to are very essential for the fungi. So it's not that easy for the fungi to adapt them. And this has as a consequence that up until now, there's only a low occurrence of resistance of fungi towards plant defensins. And I talked already about this high structural stability of plant defensins. And this allowed them actually to withstand for some time high temperatures or also extreme other conditions like a high pH or a low pH. So all these advantages make them very useful in certain applications. And which could they be? So focusing on their antifungal and also antimicrobial activity, plant defensins have a high potential to be used for gene editing in plant or plant breeding. They can be used as biopesticides or as pharmacological compounds. And in the next slides, I will give you some case studies and examples of this application so you can get some better insights. So first of all, plants as a target for gene editing. Now in the example that you can see here, we have a tobacco plant which contains a defensin of, we have potato plants which contain a defensin from tobacco. And when these plants are infected with Phytophthora infestans, which causing the potato leaf blight disease, you can see that they perform way better than their control counterparts. And in scientific research, a lot of similar experiments show that such a single defensive gene can control disease at commercial levels in agriculture crops. And often they respond as resistant or even much better towards the pathogen infection when you would compare it to the resistant cultivars that we know today, or even to plants treated by conventional chemicals. Of course, use of gene editing plants is at the moment a difficult topic, but nevertheless, I think in the future, it might become a suitable approach to use. Now, a more accepted application that would be useful today would be using them as biopesticides. So also here, there are already several examples that when you spray plants with isolated plant defensins, they lead to an increased resistance of the sprayed plant. And here you can see a study where you have oat plants, which are either sprayed with water or with a defensin of tobacco. And after infection by Puccinia, you can see that the plant sprayed by the tobacco defensin has less severe disease symptoms than the one compared to the water. And these are some case studies, but there's also one interesting application, and it's a method that is currently under revision for approval by the United States Environmental Protection Agency. And this is the use of spinach defensins to protect citrus plants against citrus greening. So citrus greening is a disease that destroyed millions of acres of citrus crops worldwide. And once such a tree is infected, there's no cure and it will die within a few years. The disease is furthermore even very easily spread through this little insect here, which is a kind of lice. And so the only way to fight this infection is either by using heavy insecticides or by sometimes even burning down the whole citrus plantations. And when you know that it takes 15 years for a citrus plant to mature, you can imagine how devastating this can be. Now, a promising solution came from some researchers that are using a modified citrus tristeza virus, or CTV. And normally this virus affects trees by injecting its genetic information into plants, forcing them to produce harmful components. So what did those researchers do? They actually removed all the unwanted virus genes and exchanged them by genes that are producing defensive genes. And they used then this modified virus to infect young citrus plants within the lab. So what does it result in? It results in a plant that is now forced by this CTV virus to induce the production of spinach defenses. After that, they take small pieces of this plant and they graft it on the major citrus plants that you can find in the plantation. So what is grafting? So grafting is a technique whereby tissues of plants are joined together, which you can see here. So the little pieces are inserted after removing a little bit of the skin of the plant. And like that, you can actually join two plants together and they will also grow together. And it's something that is commonly used in agriculture. For example, in with wines actually, good rootstocks, which provides a lot of nutrients, are combined with the upper part of a wine plant that is producing excellent grapes. And here you could compare this actually with a kind of nicotine patch, because these little pieces, they are producing slowly spinach defenses and they're releasing it towards here the major plant. And like this, they give a protection against citrus greening disease. And a good thing of this is that the fruits of these plants cannot be considered as GMOs, as there's no exchange of genetic material. And so it could facilitate the approval of this method and also saving the citrus industry for sure in the US. So let's move away from the agriculture applications and let's move to the medical field. And for this, plant defenses, and I will give you one example of what they could be used for. And this is here against invasive fungal infections. So what are these invasive fungal infections? So they're mostly caused by Candida and Aspergillus species. And for healthy people, they're not that much of a problem, but they are a major health problem for immunocompromised patients. So for example, cancer patients, and it can lead even to a 50% mortality rate. So why is this? Because these fungi are very difficult to fight against. First of all, they like to develop a kind of biofilm, and it's a kind of protective layer with which they surround themselves, and it's almost impossible for the antimycotics and the medication that we have. Furthermore, a lot of them have already developed some resistance against the medication that we're using today. So there's some need for new medication and antimycotics against these fungi. And what can plant defenses do? Now, it has already been shown for some of them that they're quite effective in inhibiting the growth or even in breaking down these biofilms. So here on the left, you can see actually what such a biofilm look like. So all these wires, these threads that you can see crossing is actually this protective layer that Candida is forming around. And when you apply a plant defensin towards it, you can see that the whole biofilm is gone. And this exposes against the Candida for treatment with normal antimycotics. So it's just one of the potential applications into the medical field. So of course, plant defensins could also be used as antimycotics, since for a lot of plant defensins, it has been shown that they are effective against human pathogens. I told you before that these plant defensins, they do bind to the membrane of fungi, So they could also be used in diagnostics, which means they could detect fungal infections in an early stage. About their very rigid and compact structure, since it has so much advantages, it could also be used as a blueprint for making new compounds. And last, although it's a little bit premature, but for some of these plant defensins, it was shown that they can slow down the growth of cancer cells without affecting healthy cells. So with this, I have given you some insights in the potential use of plant defensins, not only for agriculture applications, but also in the medical field. And I hope you enjoyed this presentation, and I'm happy to take any questions.
Thank you so much, Barbara, for sharing the story. Again, such a fascinating topic. The idea that plant defensins, these small molecules, have a role in development, first of all, but then that they might be used in such a broad range, going from GM approaches to actually even, pharmaceutical products for humans. It's fascinating how the applications of these small molecules could actually be very important for a future, right? For a sustainable future. So thank you very much. We will now go into a Q&A session. And first, I would like to thank our speakers again, of course. And my first question will go to, it's one of the questions I was posed to Prisca. A very good question in which somebody wondered how you see the practical application of this UV lights in the field. So would you put these UVB lights in a greenhouse close to the plants? How could this be done? Do you have any idea or opinion about this?
Yeah, so in the field, applying UV light is a little bit difficult because, of course, then the plants grow in the open. But I can now only speak for Belgium because I'm working in Belgium. Most of the strawberries are grown in greenhouses, so indoor. And there actually the application of UVB light would be basically very simple because here there are already LED lights applied to enhance the growth of plants or strawberries in general. And then it would be practically very easy to just add UVB light. There's always the concern that UVB light, as you've heard in the presentation, can affect us humans and our skin as well. So I'm not sure if the question maybe is aiming on the safety as well of the workers that then would work under these lamps. But here there's still research going on in what is the best way to apply UV light. So for example, you don't have to do it during the day. You can just add your UV light during the night. And this has even been shown to have a better effect than during the day. Or you can, for example, also apply your UV light just for 1 hour. You don't need to do it the whole day. And then this 1 hour, you can, for example, just do it at lunch when all the workers would just go into a lunch break, add your UV light, and then it's safe. So I think the practical application of UV light to strawberry cultivation is a relatively simple, easy step.
I would actually also like to add something to that because you're using, or you would like to be using UVB light. Yeah. And UVB indeed causes sunburn, but that's very like surface damage that is happening, and it doesn't really have that very severe long-term effects. It's UVA that you need to be worried about when you're talking about skin cancer, or if you're worried about premature skin aging. But in this case, you're using UVB, which doesn't go as deep into the cells as UVA would in humans.
Yeah, and then also, for example, I work with UVB light as well, and I, of course, have to be, well, my lamps are not hanging on the ceiling, but I'm working in a small build-up vertical farm, so I'm basically, my hands go under the light, but you can just put on gloves. And for example, your eyes, which are the most sensitive part of your face for UV light, the UV light doesn't penetrate normal glasses that I wear. Or if you would be a person that doesn't need glasses, then you can also just put on glasses to protect yourself.
Okay, thank you for that answer already. That was actually quite insightful. I'll move to Gwen now. So Gwen, I have a question for you. Is that you gave a lot of examples of these plant-own made pesticides that we actually as humans then like to eat. And I was wondering, it's like, do all these plant-made pesticides have a good taste, or were they really selected to taste good, like the vanillin? And how do I see this?
So actually, in the foods that we eat, the ones that in the crops that have been domesticated, it's usually the opposite. Many of these pesticides have a bitter taste, and because of that, we've actually lost a lot of it, because when plants were selected, or like interesting traits were selected in the past 10,000 years. So if we think back of the webinar session of last week, actually what people did, what farmers did, what breeders did, is try to get rid of the bitterness. And unfortunately, because of that, a lot of the, let's say we did not eliminate any of the metabolites, but the concentrations have dropped.
Okay, yeah, that's also, thank you.
Maybe just as an example, if you think about, like, for instance, what makes like broccoli and Brussels sprouts, it's all really bitter. These are glucosinolates, and these have also been shown at the concentrations that they're at, but also at slightly higher concentrations to have some effects that they can help prevent the development of cancer. But unfortunately, we've lost quite a bit of it.
Okay, yeah, it's so we should eat like wild plants somehow, maybe. Then Barbara, I have a question for you. So somebody asked, what is a practical advantage, if any, of using the fences by genome editing in the plants compared to using them as biopesticides? I thought that was a very interesting question. Could you answer it maybe briefly, because you're on a short time schedule?
Yeah, not a problem. So what at the moment I would think is one of the limited factors by using plant defensins as biopesticides is their production. So as I told you, the protein structure is quite important. So when you want to produce them, you already need eukaryote species, like for example, yeast. You cannot produce them in E. coli because the E. coli cannot produce such a structure. And that's why I think maybe from an economical point of view, it's maybe not that beneficial yet to produce them in large amounts. But I also think that not a lot of research has been done about it. And yeah, I'm a plant molecular biologist, so not a chemist or not such a production specialist. And maybe with some more multidisciplinary research applications, we could develop towards such a thing. And that's of course maybe that at the moment, yeah, gene editing might be easier because you just let the plant do all the work and they will produce it in the right context. So this is at the moment, I would say, a bit where the limitation is still laying.
Mm-hmm. Yeah, the answer makes sense to me. In the end, gene editing might, of course, prove a sustainable way. At least that we in GeneSprout really believe that sustainable agriculture could be done by gene editing, right?
Yeah.
Now, before you go, I will end the Q&A here because we have a very limited time schedule. But if you have more questions, please feel free to send them to GeneSprout using different platforms. I will show them later, and then we will try to answer them afterwards. We were very happy to do this, actually. So I have this. Okay. So, and then before you go, I would like to introduce you to our next and last webinar of our series, and that is actually next week. Our series is titled Plants for Human Health, the next episode, and this will be on Tuesday, the 22nd of December at 5 p.m. European Central Time, so the same time as now. So this presentation will again be hosted by ASPB, and will take place on 22nd of December. Here we will zoom in on how plants can contribute to our health. We will explain how new plant breeding technologies can help in providing everyone with enough good quality food while making agriculture more sustainable, in our rapidly changing world on the other hand. So as said, you can find us on different social media platforms. Please do not hesitate to contact us or follow us. As said, you can also pose your questions here and we will very gladly answer them. So thank you all for joining today. Thank you ASPB and Plantai, and I hope to see you all on Tuesday, the 22nd of December. Thank you very much.